Hi, good afternoon everyone. Today I will be going live for biology. Please send your questions at spmflix.com or SPM Facebook Messenger, and I will try to answer your questions within 2 hours. Again. Continue. Okay. So I know that most of you are going to have your mid-year exam. So if anything, any topic you want to revise, do feel free to send it to me that we revise it together.
Okay, cikgu boleh continue dengan apa topik dulu.
Okay. For mid-year exam, let's say we are now in around May, our target is the mid-year exam. So for mid-year exam, you will have 3 papers for biology. You have your paper 1, paper 2, and paper 3. So most of you will cover until chapter 3 or might be chapter 2 for your Form 5 syllabus. Okay, so anything about Form 4 or Form 5 that you think is important topic for your exam, okay, please let me know that we can revise it together. Okay, maybe let's say one of the popular topic for exam is something about the cell division, mitosis, or maybe meiosis. Okay, maybe at this time we can talk about cell division, mitosis. Okay. Okay, mitosis normally will come out in paper 2 for the structure, untuk soalan struktur. Okay. For mitosis, the basic concept will be like from one diploid cell, then it split, divide, membahagi kepada 2 sel anak. Then you will get 2 new cell. And for these 2 new cell is still diploid. This one is mitosis. Okay, and then for meiosis, altogether there are 2 cell division for meiosis. There are meiosis 1 and meiosis 2. So also start from a diploid cell just like mitosis. It will first divide through the process or the cell division is called meiosis 1. And for meiosis 1, the result you will have 2 haploid cells. So this is the different thing compared to mitosis. Because for meiosis, the word meio means it can reduce the number of chromosome into half. So from 2n become n, from diploid become haploid. What do you mean by diploid become haploid here? Let's say for human, we have total 46 chromosome. Every single cell in the body, we have 46 chromosome. So once become haploid, means it become 23 chromosome. So it's about half compared to the original number of chromosome. So diploid become haploid is actually meiosis 1. And why meiosis 1 is so special about this one? Because meiosis 1, for those who can recall your Form 4 syllabus, there's a phase called prophase 1. Okay, so for prophase 1 here, there's something called crossing over. They can produce variation. Okay, one more is meiosis 2. Then it will form total 4 haploid cells. Okay, so this is the brief introduction between the differences between mitosis and meiosis. Okay, now let's back to the student question. Okay, for number 1 here. Okay, teacher, can you go quick review about chapter 4, Form 4, of course. Okay, chapter 4, Form 4 syllabus, I think the topic it should be composition, chemical composition of the cell. So for this chapter, you have to know something about carbohydrate, number 1. Number 2 is about the protein. Number 3 is about the lipid. So these 3 as the main one for chapter 4. And the last one, it will be enzyme just the topic enzyme. So enzyme include actually enzyme formation, enzyme characteristic, and also lock and key. Okay, we go through maybe one by one. We start from the carbo first. Okay, for carbohydrate, we have the one called monosaccharide. We have monosaccharide, we have disaccharide, we have the polysaccharide. Normally for SPM exam, they will ask about the example. They're under monosaccharide. Example, under the disaccharide. Example, under the polysaccharide. For example, let's say mono, we have glucose. Beside glucose, we have also galactose. Beside galactose, we have also something called it's something called fructose. So again, I write here, it's better. For monosaccharide, I put here monosaccharide, we have glucose, we have fructose, and we have galactose. Okay, then for disaccharide, we have maltose, we have sucrose, we have also lactose. Okay, then for polysaccharide, we have starch, cellulose, and glycogen. So number one is about this example for chapter 4 is under carbohydrate, and the basic elements that form carbo, answer is C, H, and O, means carbon, hydrogen, and oxygen. Okay, so basically for carbohydrate, they ask about the example here and also elements that form carbo. Then another thing they can ask is about the condensation and hydrolysis. For example, glucose plus glucose here can form, the answer is maltose. And this process is called condensation. And here should add water because condensation produce water. If you reverse the arrow, then this reaction is called hydrolysis. Okay, this is the one for carbohydrate. Okay, now let's move on to protein. So for protein, you have to know the basic monomer that form protein. Answer is amino acid. Okay, let's say this circle represents one amino acid molecule. Then there's another amino acid molecule. When two amino acid molecules join together through condensation, then it will form this structure is called the dipeptide. Dipeptide. The chemical bond between amino acid molecules, this is called peptide bond. Okay, of course, when many amino acid molecules they join together, it will form the long chain of amino acid. This is called the polypeptide, or you can put here answer protein. So when it comes to protein, there's another thing you have to know for exam is about the protein structure. The protein structure, okay, let's say we have the one called primary structure of protein, secondary structure of protein, tertiary structure of protein, and quaternary structure of protein. For the primary structure of protein, means it's a linear form of amino acid. Means amino acid appear in linear line. The circle one present amino acid. So they appear in linear sequence. This is the primary structure for protein. Then when come to secondary, answer can be alpha or beta structure. So you can say alpha is a twisted form, beta like this, zigzag, zigzag. So one is alpha, one is beta. So this is for secondary. Then when it come to the tertiary, answer will be alpha plus beta, alpha, beta, and mixed together. And this will appear in actually the 3D form. like a bit round like a ball in a 3D form is the tertiary. So when just the linear sequence is called a primary structure of protein. Then when alpha, beta come together, it will be the tertiary. And when got few tertiary structure, alpha, beta, one tertiary, then another alpha, beta, then mix, alpha, beta, here also alpha, beta, then mix. And then when these four, they are linked together by the chemical bond as one protein molecule, this more complex structure, this is the tertiary. And the example in our syllabus is hemoglobin. This is quite popular for our exam. In case they ask about example for quaternary, so answer must be hemoglobin. If they ask about the tertiary structure example, then answer can be enzyme. Most of the enzymes, yes, is tertiary structure of protein. Antibodies. Okay, and one more answer is hormones. Majority hormones, also tertiary structure. Then for the secondary structure, example is our hair, our nail, fingernail, these two for our tertiary, for our secondary. Then for primary, there's only one example based on our syllabus is the insulin hormone. Okay, so this is the thing that we need to focus about amino acid. Okay, then for the next one, okay, we come to lipid first. For lipid, okay, for lipid, there are 5 different types of lipids. We have the one called wax. We have the one called fat, wax, fat, oil, phospholipid. And one more here, 1, 2, 3, 4. Okay, we have fat, oil, wax, phospholipid, and here should be one more here. Okay, is steroid. Okay, so there are 5 altogether. Normally popular for exam, answer fat and oil. Because fat and oil, the molecule, they are about the same, is called the triglyceride. Means with 1 glycerol plus 3 fatty acid linked together. Like this. They will link together one. Okay, so for this one, is very popular for exam. So please focus on this one. Okay, if something come out can be like this. Let's say 1 glycerol plus 3 fatty acid. And form here, okay, let's say this way first, form here, answer is 1 triglyceride molecule. And here, we'll plus another 3 water molecules. So this process is called condensation. If reverse, is called hydrolysis. Okay, so this part is quite popular for exam. So with 1 glycerol plus 3 fatty acids can form 1 triglyceride, means this actually fat. Okay, and 3 water molecule. In case they show you the fatty acid got 1 double bond, 2 lines, double bond, or here double bond, or here double bond. Once got double bond, it's no longer fat. This is actually oil. Okay. Okay, so this one for lipid. Next, we move on to enzyme. So about the enzyme, okay, for number 2 here, there's one student, Ryan, asked about, okay, can you explain on the mechanism of enzyme action? Of course, yes. Okay, for mechanism of enzyme action, so we will link to the lock and key hypothesis. About lock and key hypothesis, we all know that enzyme reaction, they are highly specific. Means one type of enzyme can only react with one type of substrate, one type. So it's a 1-to-1. Example, our salivary amylase can only digest starch. Our maltase enzyme can only digest the maltose sugar. So it's 1-to-1, is highly specific. And for this highly specific mechanism can be explained by a hypothesis. It's called lock and key hypothesis. Okay, this is one. Lock and key hypothesis. Or you can put here lock and key mechanism. So for lock and key here, it shows that the enzyme represent the lock. Enzyme is the lock. And then the substrate is the key. Okay, I put something like E here, represent the enzyme. This is the substrate. So the way they react, you must look at this one, the specific part here that fit the substrate. I use here red color for you. This is called the active site. Okay, make sure that the substrate, this structure, and the active site of enzyme, they are complementary to each other, means can fit, like lock and key. Okay, so like lock and key. Okay, for example, for why do you use the word lock and key here? Let's say for your house, you have a lock there. So you can only use your house key to open the lock. You cannot use other key to open your house lock. So basically, it's very specific, it's 1-to-1. So make sure that it fit exactly the thing here, they only can go in and got reaction. So when enzyme and substrate combine together, it will form the structure this is the enzyme. And then for the substrate, sorry for the same color here. Okay, so we join together. When they join together, this is actually called enzyme substrate. Enzyme substrate complex. Okay, once in enzyme substrate complex form, what happened here is reaction start. And at the end of this reaction, enzyme will remain the same. Means before, after is the same structure, no change at all. But only for the substrate here, I will break into two. And this is the product. So actually, after it formed the product will leave the active site. And now enzyme is considered free to bind with another substrate to continue or to repeat the same reaction here. So basically from lock and key hypothesis, got two main points you must know for exam. Number one, lock and key hypothesis explain that the enzyme reaction is highly specific. Make sure that the substrate can fit into the active site, then only got reaction. This is number one, specific. Enzyme reaction is highly specific. Number two, at the end of the reaction, if you notice, only the substrate turn into product, but the enzyme remain the same. Before, after, no change at all. It means enzyme can be reused. Enzyme can be recycled, can be reused. So normally enzymes just needed in small quantity. Okay, let's say if exam give you the lock and key mechanism about enzyme, ask you based on this lock and key diagram, okay, this like this lock and key diagram here, state two characteristic of enzyme. One, enzyme reaction is specific. Then for number two, okay, enzyme is not destroyed at the end of the reaction. So only got 2 points. How about like this? If I mention based on lock and key hypothesis, enzyme is sensitive to temperature. The optimum temperature for enzyme is 37 degrees Celsius. If you put optimum temperature or optimum pH for this one, so basically you cannot score because lock and key hypothesis never explain temperature, never explain pH value. It only explain the main thing is enzyme and substrate. They can react together and must be very specific. Means make sure that the substrate structure can fit in the enzyme active site. They only got reaction. It means it's specific. And number two, enzyme is not destroyed at the end of the reaction. So only these two main points you can mention about enzyme characteristic based on the lock and key hypothesis. Okay, so what else we need to know about enzyme for exam? Okay, besides lock and key hypothesis, another one you have to know is about the enzyme formation. Okay, this one to link your actually chapter 2 about the organelles linked to the enzyme formation. Okay, let's say this is one animal cell and this is the nucleus, let's say. Then this is the rough ER. And then we've of course got ribosome attached to the rough ER. Then we have the Golgi apparatus. Okay, Golgi apparatus. Okay, inside let's say with the DNA, the double helix DNA. Okay, let's imagine about the enzyme formation. This one can come out, let's say, in structure or maybe in assay. Okay, step number 1, DNA, the genetic information from DNA is copied into RNA form. Then the RNA can leave the nucleus. RNA can leave the nucleus across the membrane and come to the ribosome. We all know that ribosome is site of protein synthesis. So after ribosome receive the information from the nucleus, which is RNA, then protein is synthesized. Site of protein synthesis for ribosome. Then protein that synthesized by ribosome will be transported by this structure is called rough ER in the form of vesicle. And this vesicle inside is the protein that produced by ribosome, all the way sent to the Golgi apparatus. And this vesicle, the name is called transport. Vesicle. Why the name is called transport? Because just to transport the protein from ribosome all the way to the Golgi apparatus. So when it comes to the Golgi apparatus function is to modify or can say process protein into the enzyme. Then the enzyme is packed in also vesicle form and sent out of the cell. Of course, the enzyme that sent out of the cell digest the thing outside the cell. This is called extracellular enzyme. Enzyme and it's extracellular enzyme. Okay, so basically for chapter 4 about enzyme, what you need to know, number 1 is the enzyme general characteristic. Okay, we revise here one round. Enzyme is biocatalyst, number 1. Then for number 2, enzyme is actually protein. Okay, only living organism can produce protein. Then for number 3, enzyme, yes, is very sensitive to temperature and also pH. Okay, an enzyme reaction is highly specific. Enzyme can be reused. Enzyme is not destroyed by the reaction. But when it comes to the lock and key hypothesis, we can only say enzyme reaction is highly specific, number 1. Then for number 2, enzyme is not destroyed at the end of the reaction. Other we cannot mention based on the lock and key hypothesis. And enzyme is very popular for paper 2, for structure and also assay. Lock and key can be both assay and structure. So please prepare. Okay, for the next one, number 3, okay, by Farah. Okay, hi. Okay, what is the function of cilia? Okay, so basically cilia is the short hair. Short hair, so they can move. Boleh gerak, can move one. So basically cilia is for movement. Cilia is mainly for movement. Just that short hair can move. For example, along the esophagus, okay, they got the movement here by the cilia also, and also the peristalsis. Let's say for the girl fallopian tube, also got the hair to move the egg cell. So basically cilia is for movement. Okay, along the trachea here, we also got the cilia also. So I think the popular one for SPM is the trachea part. So the trachea here, got the cilia. So basically is to move the mucus, is to remove the mucus. If based on our syllabus, cilia, they focus more on the trachea part. Let's say, this is the trachea. Okay, trachea. Then to the bronchus. Here is also the bronchus here. Then of course we have the lungs. Inside we got many alveoli. So the lining here can secrete mucus. This lining here can secrete actually mucus. Mucus, the sticky fluid. The purpose is to trap the dust. In case got dust, you inhale, got dust, so the dust can stick to here. Purpose, so that it won't direct go into the lungs and cause infection. After here trap the dust, the mucus is still in your trachea here. Okay? What you can do next is the cilia. Here the short hair cilia. So imagine cilia is short hair. So for cilia, what they can do? They can move. They can move like they can move the mucus together with the dust or bacteria, move and all the way come out here. So that you feel like the thing won't go in direct, will come out here. Of course, then the mucus will go to here also, right? Go to your mouth. So at that time, you can decide you want to swallow it back, the mucus, into the stomach, or you want to split out. Okay, this is your choice. Okay, one soalan came back from here, from SPM one. Let's say for those smoking one, the heat from the smoke may able to destroy the cilia along the trachea. Once destroy the cilia here, when the cilia is destroyed by the heat, imagine the heat like the fire burn the hair here, destroy the hair here. So the hair then cannot move anymore, cannot beat anymore. If cannot beat anymore, then how to remove the mucus come out with the bacteria so that it won't go into the lungs and cause infection? Okay, answer is for those heavy smoker. So normally they got this habit is they will cough like so use the pressure to force the mucus to come out. Because the thing stuck there cannot be moved by the cilia, you don't feel comfortable. So this effect of smoking may destroy the cilia here. Okay. I hope that this one can let you understand better about cilia. So basically cilia is just a very short hair, basically for movement. Okay. Example, along the trachea here is for defense system. Okay. For number 4. Okay. This one is about, okay, reflex action. Ali's finger accidentally touched, okay, hot object. And please explain briefly how his reflex action function to avoid injury. Okay, 3 marks all together. I think this one should be from book. Okay, okay, like this. Let me explain to you about reflex first. Number 1, you must have the basic understanding about reflex. This Form 5 syllabus, Chapter 3. Reflex number 1 is controlled by the spinal cord. It's not controlled by the brain. That's why it can be very fast, okay, very fast response, and it happens without your conscious control because the whole thing is controlled just by the spinal cord. Number 1. So spinal cord is the control center for reflex. For the hot object or even sharp object will be the same answer. It involves altogether 3 types of neurons, which are afferent neuron, efferent neuron, and the interneuron. Okay, so we have afferent neuron, efferent neuron, and the interneuron. So the control center is the spinal cord. Okay, so of course for this case, your answer should start from the receptor. Okay, receptor here. Okay, example, this is receptor. This is, let's say, is hot receptor. Can be hot or can be heat receptor. Detect hot stimulus. Okay, so hot stimulus is detected by hot receptor. And then impulse is triggered by hot receptor and sent all the way to the spinal cord. Okay, for the spinal cord, be something like this. Okay, spinal cord here. Okay, come back to this one. All the way that go in, this is actually the afferent neuron. The impulse go in here, impulse direction. Okay, from the hot receptor to the gray matter. This is the middle here. This is called the gray matter. The whole thing here is the gray matter. Okay, gray matter of spinal cord. And this one is the afferent neuron. Afferent neuron. So I repeat here, step number 1, you have to mention, okay, this actually talk about the hot receptor here. So hot stimulus is detected by hot receptor. From here, you will get 1 mark. And then hot receptor trigger impulse and send by afferent neuron to the gray matter of the spinal cord. So afferent neuron will give you the second point here. Then in the gray matter of the spinal cord, impulse is transmitted to interneuron. For this case, involve interneuron. So interneuron give you another 1 mark here. And then from interneuron, impulse is transmitted to efferent neuron. Of course, efferent neuron carry impulse away from the spinal cord to effector. Okay, let's say for this case, effector, possible answer, is your biceps muscle. Okay, because for this case, effector is the muscle. Let's say we talk about your finger touch a very hot object, then you will withdraw your hand immediately and it's automatic. So for this movement, of course, involve the muscle is the bicep to contract. Okay. So your bicep response is contract. Okay, once involved here, muscle contract, then respond is you move your hand away from the hot object. So when come to exam, okay, one more time here, 3 marks for this one should be structure, but answer is about 5 points altogether. So when come to exam, any 3 points you mention for this one, you can also score maximum 3. I repeat here, number 1, hot stimulus is detected by hot receptor. You will get the first point. Then hot receptor is triggered to produce impulse or to generate impulse. Means hot receptor trigger impulse formation and impulse is sent by the afferent neuron. Afferent neuron is your second point. One more name for afferent neuron is called sensory neuron. You can put afferent neuron or sensory neuron, they are the same. You will get a second point here. Then all the way in the gray matter of the spinal cord, impulse is transmitted to interneuron, point number 3, to efferent neuron, point number 4. Then efferent neuron send impulse to bicep to stimulate bicep to contract. So bicep contract is response. You get here 1 point. Contract. Of course, you can mention also, then the hand move away from the hot object. So total there are 5 points to explain about the reflex action. Okay, I repeat here, no matter it's hot object touching a very hot object or very sharp object, yes, you will have the reflex. And for this reflex, you will have total 3 neurons to help you to respond. Afferent neuron means sensory neuron, interneuron, and efferent neuron. One more name for efferent neuron is called motor neuron. They are the same. You can put efferent neuron or motor neuron and send impulse all the way to effector to respond. And for this case, the effector is the bicep. Okay, normally it's muscle here because for external stimulus. Okay, I can give you a bit soalan kebab about here. Okay, let's say if someone experienced very serious, let's say car accident and injured, this one is called the dorsal root. The whole thing here, let's say no more, gone, damage. Okay, serious damage here. If like this, impulse cannot flow all the way to the spinal cord anymore. So if this one happened here, totally damaged, when touch a very hot object here, still got reflex or no? Answer is no reflex. Why no reflex? Because impulse cannot be sent to the spinal cord by afferent neuron. So that's the answer. How about like this? If the accident only damage is the ventral root at the bottom here, the one that store the motor neuron, let's say this one is perfect, normal, nothing happened, but only the ventral root is seriously damaged, let's say during accident time. When damaged here, then impulse cannot be sent all the way to the bicep, means also got no response. So basically for this case also no reflex action. But the difference is impulse still can all the way go into the spinal cord. It means you can feel the pain. You feel it's hot, but you cannot respond because the motor neuron is not functioning. Okay? So this is the one that involved in reflex action. Okay, in case you want to know more about reflex, there's one more type of reflex is in your syllabus. Okay, it's the knee jerking reflex. For that one, it's a bit special. Everything look like the same, but for that one is you have to remove the interneuron. It means from the afferent neuron, direct connect to the efferent. The gap, you make it smaller. Okay, the gap, you draw it, make it smaller. Something like this. This is the afferent neuron. Draw, make it longer. And then here also, the motor neuron. Make it join together, come closer. And the gap here, of course, is called synapse. Okay, so for this case, is for the knee jerking experiment. So all this no, all this no. For the knee one. The one is for the special case, only involve 2 neurons. Okay, so next one. Okay, for number 5. Okay, how to answer essay question in bio exam. Okay, this one must depends on what type of essay that you get. Okay, for essay question popular for bio, number 1 is about ecosystem. It's about pollution. So for pollution, there are few popular topic for essay. One is the greenhouse effect. You have to explain, okay, the cause of greenhouse effect. Main point is the release of carbon dioxide. And what can release carbon dioxide? From burning. Forest burning, open burning, from petrol, from car burning. Okay, yeah. So basically all this, yes. And you have to explain the process. Okay. How can cause the trap the heat, the global warming, and then about the effect, and also how to overcome the problem. Okay. And then beside the greenhouse effect, also can be eutrophication. It's under water pollution there. Okay, and then ozone layer depletion. The main cause is really CFC. So for essay, of course, depends on how many marks given. If for the 10 marks essay, you have to write minimum is 10 points. When give you the 10 mark essay, marking scheme will have more than 10 points for you. You can write any 10 points correct from the marking scheme. Let's say 10 marks essay, max already. Maybe actually marking scheme, they will have 15 to 18 points. So you write any 10 points from there, you also can score maximum 10. but some essay maybe give you only 5 marks or maybe only 8 marks. So during the one 8 marks means the one 8 points also. So just try your best, okay, to get all the points. And how to write a very good essay is you must give a bit introduction, like you write the essay, normal karangan, okay, for your English or BM, and then you start your point, okay. You can also put in paragraph form. Let's say I want to explain one big point. I put in one paragraph, okay, one main point, and then my explanation to that point is one paragraph. Then another point, I explain the point also, the main point, in another paragraph. When you put in paragraph form, it's easier for the marker, means for your school teacher to mark your answer because it's very well organized. So for the good essay thing is, okay, you must try to imagine you are writing something to show your friend who are not studying biology. Let's say from art stream student, if your friend read your essay, they can understand what you're going to say. Your essay is very good, number one. And for biology essay, you have to put in all the specific keywords that in biology in your textbook. Because if without the keyword, it's very hard for you to score. Because biology is science subject. We have the specific keyword for certain topic. So the keyword must be there, number one. Number two must be in complete sentence. Okay, yeah, for essay, please write in complete sentence. Point form, yes, you can write, but must be also in complete sentence for essay. Okay? Okay, so we move on to the next one. Okay, for number 6, teacher, can you explain the oogenesis? Okay, this is actually chapter 4, form 5, or maybe some school cover this one for the mid-year. Okay, so for oogenesis means ovum formation. Oogenesis is ovum formation. Okay, like this. Basic intro first. Oogenesis, the word oo means ovum. Genesis means formation. So combine, oogenesis is ovum formation. And in which part of the body can happen ovum formation must be the girl sex organ. It's in the ovary, number one, and continue will be in the fallopian tube. Yes, you hear me right? I repeat here, oogenesis occurs in ovary for the first stage and might also continue to happen in the fallopian tube. Yes, it's in ovary, the sex organ, and then continue in the fallopian tube. Okay, why like this? Okay, let's start now. For the oogenesis, of course, start in the ovary. Start from a diploid cell. The inside the ovary is called a primordial germ cell. Then for this primordial germ cell, it can perform repeated mitosis. Repeated mitosis to form many diploid cells. The original diploid cell in ovary that can form ovum eventually is called primordial germ cell. Primordial germ cell, original diploid cell in ovary. Then after many times mitosis should be repeated, they can form actually many diploid cells. So mitosis in the sex organ, the purpose is to increase number of diploid cells. Get more diploid cells first, then only perform meiosis. Okay? And then from here, let's say this is a cell that eventually can undergo meiosis. So before meiosis, it will undergo the process is called the growth. Okay, it's something like the interphase, the G1 phase, the S phase, and the G2 phase. It will grow to the maximum size. Then it start to divide through meiosis. Okay, meiosis actually got 2 cell division. We have meiosis 1 and the meiosis 2. So for meiosis 1 in girl is a bit special. How special? Meiosis 1, normally for cell division, the cell will divide equally into 2 new cells. Should be the same size. But for girl, it's special is meiosis 1, it will divide unevenly into 2 new cells. Will be one bigger and one smaller. And the bigger one is the one that we call egg cell. In BM ialah sel telur. The big one and the small one. The big one is the egg cell. After meiosis 1, can form the egg cell direct. Okay? And then for the small one, is called polar body. Okay, one more time here. For G, okay, oogenesis means ovum formation. Okay? Start from original diploid cell in the ovary is called a primordial germ cell. And then it will perform repeated mitosis. Should be many times mitosis, repeated mitosis to form many diploid cell. And for this diploid cell is called oogonium. This is called oogonium. Oo means actually it's ovum egg cell. Then it's called gonium here. Oogonium. The name is called oogonium. When got many, it's called oogonia. If many, it's called oogonia. Means for plural. If one, it's called oogonium. When grow to the maximum size just before meiosis 1, is called primary oocyte. Okay, again, the oo means egg cell. Okay, the ovum here. Cyte means the cell, like the egg cell here, primary oocyte. Then the egg cell, scientific name for egg cell is called secondary oocyte. Secondary oocyte. So we have the primary oocyte followed by the secondary oocyte. And secondary oocyte is the one that we call egg cell in general. Means when come to here, the whole thing happen in ovary. Everything here is still in ovary. So from the primordial germ cell all the way until the egg cell form, everything happen inside the ovary. Okay. Then only when come to, okay, let's say for girl, they will have the period cycle. So during the period cycle means the menstrual cycle, on day 14 of the cycle, you will have a day is called the day of ovulation. So the ovary will release one egg cell into the fallopian tube. If this egg cell in the fallopian tube got sperm enter and penetrate the egg cell, then only the egg cell will perform meiosis 2. Understand or not? So again, from ovary on day 14 of menstrual cycle, it will burst and release one egg cell out. The egg cell is here, the secondary oocyte. So after meiosis 1, form the secondary oocyte, means the egg cell. This egg cell will come to the fallopian tube. Okay, come to the fallopian tube. In case got sperm enter and penetrate the egg cell, then only this egg cell will perform meiosis 2. So meiosis 1 is in ovary. Meiosis 2 is actually in the fallopian tube. Okay, in case got sperm, let's say got sperm, okay, want to penetrate the egg cell, then in the fallopian tube, here perform meiosis 2. When come to meiosis 2, will be the same thing. The egg cell will divide unevenly into two. One bigger size, one smaller size. The bigger one is the actual ovum. The smaller one is just called polar body. All this also called polar bodies. Okay. So the first one here is called polar body. Then bottom, these three also called polar bodies. So any difference? This is called first polar body. The bottom one answer is second. Polar bodies got three here. So only get one ovum. Okay, like this. From here, one ovum. Let's say this is the sperm. Okay, the tail remain outside. The nucleus go in. The haploid nucleus go in. This is a haploid nucleus for the ovum. Then these two nuclei will fuse together and form zygote. The diploid zygote means form baby. Okay, yeah. So will be like this. For girl, it's quite special one. Oogenesis cannot complete inside the ovary. Okay, can only complete until halfway, until meiosis 1, until actually the egg cell. And then it will be released into the fallopian tube on the day of ovulation. Okay, then if got sperm penetrate the egg cell, the secondary oocyte, then only this egg cell will perform meiosis 2 and form the actual ovum. Once form the actual ovum, then only the nucleus of the sperm and nucleus of the ovum, they will like nucleus of the sperm, nucleus of the ovum, they will fuse together, N plus N become the diploid zygote. Okay, yeah. So the ovum is an intermediate between the egg cell and the zygote. The intermediate is the ovum. If no sperm, for girl you won't perform meiosis 2, there's no ovum form, only egg cell. In case got no sperm enter your body at all, okay, then answer is meiosis only until 1, then here stop. You will have no meiosis 2 at all. The egg cell just come out together with the menses during the next menstruation. Okay? Okay, so this is the thing for oogenesis. Okay, hope that it will help you in your exam. Okay, for number 7. Okay, this student asked how to differentiate pancreas and liver in regulation of blood glucose level. Yes, both pancreas and liver organs both also regulate our blood sugar level, our blood glucose level. The function of pancreas only one thing: secrete the hormone. So what hormone can be secreted by our pancreas organ? Got two hormones, insulin and glucagon. So basically, how about the liver? Liver, they don't produce any hormone. Liver just provide the place for the sugar to be converted into glycogen. Means the conversion between glucose and glycogen happen in the liver. Okay? But for this conversion to happen in the liver, they need the order. The order is actually the hormone, insulin, and glucagon. Who produce this hormone? Answer is the pancreas. So pancreas produce hormone, carried by blood all the way through to the liver. So in the liver, they perform the function. So basically, pancreas is the hormone gland, is the endocrine gland, and liver is the target organ for this case. Example, let's say this is our liver. Liver. And then for our pancreas, so pancreas, let's say function here is to produce hormone. I give an example. Now we talk about high sugar level. Normally after meal, after lunch, okay, after breakfast, of course your sugar level will shoot up. Okay, once your sugar level shoot up, at this point, your high blood sugar level is considered as a stimulus. Rangsangan, stimulus. So stimulus can be detected by your body. And then your pancreas will be stimulated to produce the insulin hormone. Okay, the insulin hormone. So insulin hormone is then carried by your blood all the way to the liver, is insulin. I repeat here, after meal, let's say you eat 1 plate of rice, at the end you will have sugar. Then the glucose level will shoot up above the normal level. At this point, your pancreas is stimulated to produce insulin, all the way carried by your blood, all the way sent to the liver. Then in the liver, excess glucose is converted into glycogen. Okay, and this glycogen can be stored in the liver for temporary. When come to, let's say you feel very hungry, okay, very hungry here, means your blood sugar level now is considered low. When you have low blood sugar level, hungry now, low blood sugar level means your body not enough sugar. When not enough sugar, you need to convert the glycogen back to the glucose to supply you sugar so that it can produce more energy. Okay, I repeat here. About pancreas and liver, both are organ, that involve in regulation of blood glucose level. Pancreas, the main function in this case is to secrete hormones. There are two hormones can be produced by the pancreas. Number one is insulin. Number two is glucagon. Okay, so very simple. Normally, after meal, after eating, even just drink a cup of teh tarik is also sweet. So when the sugar level is high, your pancreas organ is stimulated to produce, to secrete insulin hormone. All the way we come to the liver. Of course, from pancreas, how to go to the liver? Of course, this is carried by our blood. Hormone is transported by our blood. So all the way sent to the liver. So the liver is the target organ for this hormone. Then in the liver, insulin will perform its function, which is to convert excess glucose. You must say it's excess glucose, because the word excess means extra one. You only convert the extra sugar in your blood into the glycogen form and store in the liver for temporary. In case exam asks you, beside your liver can store glycogen, name one more part in the body can store glycogen. Answer is your muscle. So actually, our textbook mentioned mainly is the liver store glycogen, but muscle also can store glycogen. So when you feel very hungry, the glycogen will be converted back into sugar for you to burn and get energy. Okay, so basically, the difference here is just liver is for the conversion, and then for the pancreas is a secret hormone to control the whole thing. Okay. Okay, so we move on to the next one. Okay, by the same student, how to differentiate afferent neurons, interneurons, and efferent neurons. How they are linked together. Okay, like this. Okay, let me explain to you this one. Okay, I use the reflex action for you to understand better, link for you together. So this is, let's say, the spinal cord, the gray matter. The outer part is the gray matter. Sorry, it's the white matter here. Okay, for the afferent neuron, okay, for the afferent neuron must be from the receptor is the nerve. Actually, neuron means nerve cell. Neuron, one neuron means one nerve cell. So when you put neurons, got S, means nerve cells, means nerve tissue. Okay. So for the afferent neuron, must be from the receptor all the way you draw all the way in, and then ending you draw like this. This one represent the axon terminal. And for the afferent neuron, very special characteristic is the cell body. The cell body is located here. This is the cell body. Cell body. Compared to motor neuron, let's say. For motor neuron, the cell body is inside. And then all the way come out. We'll come to the effector. This is the effector. Can be your muscle, can be your endocrine gland to respond. Okay, like this. One is output, one is input here, the impulse. Okay, please focus on the cell body. About the cell body here, for the afferent neuron, cell body is at the middle of the axon. This long structure, the long structure here, this is called axon. Like at the middle, like next to the axon is the cell body. And for the motor neuron, cell body is at the end or at the beginning of the nerve. So this is the axon at the beginning. This is at the middle. So from here you can see, okay, one is the afferent, one is the efferent neuron. And for afferent and efferent neuron, when it comes to more special, both also got myelin. But for the interneuron, interneuron is the one that without the myelin, without the fat layer, protect the axon 1. You can see no myelin at all, automatic answer is the interneuron. If got myelin, please focus on the cell body location. If the cell body, let's say this one is the axon, cell body located at the end here must be motor. If the cell body at the middle like this, must be answer the efferent. So I repeat, if cell body, the nucleus, cell body is at the end of the axon, the cable, the nerve here, at the end here got the cell body nucleus here, this is the motor. If at the middle, next to the axon only, answer is the efferent. If for the axon got no myelin at all, no fat at all one along the axon, the answer is the interneuron. Interneuron is very special. Interneuron is only located in our CNS, which is central nervous system, the brain and the spinal cord. We have the interneuron. For our arm, leg, all this, we have no interneuron. So interneuron only for the CNS, the middle one, the brain and the spinal cord, we have the interneuron. So normally interneuron, the name inter means in between. This one interneuron must be located between afferent and the efferent. So the input afferent, the output efferent, in between is the interneuron. So the name is between them. So it's called interneuron. Okay, yeah. So for most of the response, we have the interneuron involved. Once the impulse all the way sent to the spinal cord, it will pass to the interneuron. Then through interneuron, will send all the way up to the brain here, let you process the data, let you know what happened outside, let you think and let you respond. Then you will send the impulse all the way down back to the spinal cord. Everything is interneuron. Then when the impulse come up from your spinal cord to your arm or to your leg to respond, then come up is the efferent neuron. So efferent neuron is to send impulse from the CNS. Efferent neuron means the motor neuron is to send impulse all the way from the CNS to effector. Okay, I put it this way in summary for you. In terms of function, they are all neurons, no matter interneuron, afferent neuron means the sensory neuron, or the efferent neuron, motor neuron, they are all neurons. Neurons means nerve. Nerve is to send impulse. So for all three, same function, transmit impulse. Just that transmit impulse from where to where in the body, different location. For the afferent neuron, when it comes to exam, if they ask you, state the function of afferent neuron, then answer is to transmit impulse, means to send impulse, to transmit impulse from receptor, your sensory receptor, to the CNS, central nervous system, can be the spinal cord, can be the brain direct. Okay, this one for the afferent neuron. Send impulse, transmit impulse also can, from receptor to, let's say, the spinal cord. Then for interneuron, is to pass the impulse, is to send the impulse from afferent neuron, pass to the efferent neuron. Of course, exam time, you cannot say pass the impulse. You must say transmit the impulse. Keyword, transmit the impulse. So for interneuron is to transmit nerve impulses from afferent neuron to efferent neuron. Okay? Then for the efferent neuron is to transmit impulse from, let's say, spinal cord all the way to effector. So to effector, to let you respond. So efferent neuron, one more name is called motor, because it will send impulse to your, let's say, body to move like motor. So one more name for the efferent neuron is called the motor neuron. Okay. Okay, maybe I give you the summary for this one again. Okay, about the interneuron in terms of structure, is very special, got no myelin at all. If no myelin, direct answer is the interneuron. If the diagram show you myelin, okay, so can be afferent or the efferent, got two possible answer. Then how to differentiate these two? Look at the cell body, the nucleus location. If the cell body is located at the end of the axon, or at the beginning of the axon, this is a cell body here, then it's the motor neuron. efferent. If the cell body at the middle here, like this, suddenly come out the cell body like this, okay, this is the efferent neuron, the sensory. Then in terms of function, they are all the same. Carry impulse, send impulse, transmit impulse. Of course, the best term to use is transmit impulse, means send impulse. Just that from where to where, different location. Okay. Normally, some will link to reflex, and you have to draw all the afferent neuron, interneuron to the efferent neuron, will link all together one. Okay. Normally, it's for reflex action or for the normal response, but for SPM, it's more on the reflex action, link all three together. Okay. Hope it can help you. Okay, for the next one, how the antagonistic muscle work. Okay, for antagonistic muscle, okay, the popular example, let's say for human body, is about your biceps and your triceps muscle. Antagonistic muscle means refer to a pair of muscles, one pair of muscle. One pair means got two. One pair means got two. So normally for our body, the whole body is also antagonistic muscle. Based on your Form 5 syllabus chapter 2 here, locomotion and support, antagonistic muscle, okay, let's say for human body, the popular example is your biceps and your triceps. Let's say when you bend your arm, okay, which muscle that contract, your biceps or your triceps? So when you bend your arm, of course you can feel your arm, the top here, slightly become like a bit tight, the muscle. So this muscle here contract. Then at the same time, you can feel the muscle at the bottom here. It's a bit soft. Soft. Soft means the muscle relax. So when it comes to the antagonistic muscle, you must explain antagonistic muscle is a pair of muscle. When a member of a muscle contract, another member must relax. Means when one muscle contract, another muscle must relax. Example can be like, let's say, explain, to bend the arm, which muscle contract? Answer is biceps muscle contract. And you must mention bicep contract means the tricep will relax. If you straighten your arm to the max, then will be the other way. Then your tricep here will contract. Then your bicep here will be soft. When here hard, the muscle will be soft. When here soft, means the muscle relax. Okay, why not our muscle, bicep and tricep contract at the same time? It cannot happen. Because when the muscle contract, means the muscle pulling. So when the muscle pulling, means when here pulling, your forearm should move this side because here pulling. When here pulling, the muscle contract. When here pulling, here must let go, relax, so that your arm can bend in one direction. Move like this. Okay, let's imagine when the bottom, your tricep contract, when here pulling, here let go, you straighten your arm. So it's very normal. If these two muscles contract at the same time, your arm do not move in which direction. Okay, so muscle must be one contract and one relax. Okay, beside your arm, bicep and tricep, for human body, actually the leg are also popular for exam. For your leg, when the front muscle contract, the back muscle relax. So when the front muscle contract, your leg, okay, the upper leg here, when the front muscle contract, the behind one relax, you will straighten your leg. Okay, then the front muscle contract for the leg muscle, then we straighten the leg. When the back muscle contract, then you will bend your leg. So like this, alternate to move, to walk. Okay, okay. Beside about the bicep, tricep, about the leg muscle. Okay, there's one more example under antagonistic is our human heart organ. You can recall your chapter 1. For human heart, upper part is called the atrium or the atria. Lower part of the heart is called the ventricle. So for the heartbeat, should be like doop doop. Like heart is around here, like doop doop, doop doop, doop doop. When the lower chambers contract, the upper chamber must relax. When the upper contract, lower must relax. Heartbeat like doop doop, doop doop, doop doop. This is also antagonistic muscle. Means when one muscle contract, another muscle must relax. When the upper chamber contract, pump the blood down to the ventricles. Here must relax to receive the blood. Cannot like upper and lower chambers contract together. I think if we jam here, it's heart attack. No, so one contract, one relax, antagonistic muscle. Okay, then about antagonistic muscle, if asked you to define or explain, you just say it's a pair of muscle in which when one muscle contract, another muscle relax. That's all. Unless they give you the diagram, you have to base on the diagram, the name of the muscle, you explain the movement. Okay, for antagonistic muscle, beside human, also can apply to animal. Okay, can apply to animal as well. Okay. Okay, next one. Okay, teacher, can you please explain nitrogen? I think it's not recycle, should be nitrogen cycle. Kita, nitrogen cycle. Okay, okay, so for, okay, nitrogen cycle can go to essay, total is 10 marks essay. Can be 10 marks essay, that's the max already. Okay, and also can go to the structure or maybe go to the objective. So for this about nitrogen cycle, I'll give you a bit intro first. Why we need to study this cycle? Why is this nitrogen cycle important to us as a human? Nitrogen is the element that all the living organisms need to form protein. Human, we need protein. Plants, they need protein as well. So for plant, how to get nitrogen to form protein? You must understand one thing. Plants, they are producer. Without plant, human, we can't survive. We are consumer. Animal, we are consumer. Plant, they are producer. So main point is plant can get nitrogen to form protein for them to grow first. Once producer can grow, then consumer can eat the plant, we can survive as consumer. So this cycle basically talk about the protein. Nitrogen is one of the very important element to form protein. Okay, okay. Any points you have to know about this cycle? Number one is all the microorganism. Altogether, there are 5 bacteria, means about 5 steps in this cycle. Every single bacteria, you need to memorize the scientific name. Some is scientific name, some is the general name. For example, we have the one called Rhizobium species, Nitrosomonas species, Nitrobacter species. So it's specific name for the bacteria. Okay, and then you have to know function for each bacteria involved in which step. And then what is the function for each one? Let's say for maybe from the gas nitrogen all the way to the ammonium that absorbed by the legume plant, what is the bacteria involved? It's the Rhizobium species. You can name Rhizobium species, you'll get 1 point. Okay, something like this. This is nitrogen gas, N2. And this is the legume plant. Okay, if you can recall your chapter 8 from 4, legume plant here, they got the root got a swollen structure, is called the nodule. Inside the nodule, live a type of bacteria. The name is called Rhizobium species. You may put SP dot, you must underline Rhizobium. No underline, then is wrong. Okay, you must underline. This actually scientific name. For scientific name, one name give you 1 mark. You must underline Rhizobium species that live in the nodule of the legume plant that can fix. Function here is to fix nitrogen. Of course, exam is to spell nitrogen here. When you say fix nitrogen, means convert the nitrogen gas into the form that can be absorbed by the legume plant. So actually fix nitrogen, it means convert this nitrogen all the way into what form? Into the NH4 plus. So this is actually the soluble form. Just now the N2, this is actually gas, nitrogen gas. This is nitrogen but in soluble form. Can you see a positive ion? Means can dissolve in water. So when the plant absorb the water, these minerals dissolve in water can go in together. The main point is plant can use this nitrogen to form protein. Want to form protein, there are 4 basic elements: C, H, O, and nitrogen. Main point is nitrogen. You can imagine plant, they can perform photosynthesis. When they perform photosynthesis, they can form C6H12 and O6, means it's actually glucose. Means sugar plus nitrogen can become the protein. But the point is, from where can get nitrogen to form protein? They need protein to form the new cell to grow. So with this nitrogen can get from here. But who convert the gas from A into the soluble form that can be absorbed by this legume plant is the bacteria called Rhizobium species. So for this step, you mention the bacteria is called Rhizobium species, spelling correct. SP dot underline, you will get 1 mark. Function is to fix nitrogen. You get here another 1. This is 1 step. Then after that, if let's say plant die, then it will be decomposed. Okay, it will be decomposed into also the ammonium form. Of course, it's also possible that maybe human, we eat the plant and then become the human protein. Okay, one day, okay, the plant die, human die here, same thing. The dead body will be decomposed by saprophytic. Saprophytic bacteria. Can be saprophytic bacteria, also can be saprophytic fungi, means the fungus, decompose the dead body and return the protein back to the soil in ammonium compound. Means in ammonium form. NH4 plus ammonium. Okay, like this. Our body, we got cell. Plant also got the plant cell. Our body cell, all these cells also got protein. So after, let's say we die one day, actually all this protein will return to soil. And this bacteria will come to eat our body. They will decompose all the cell here, all the tissue here, take out all the protein, return to soil. Return to soil and become the natural fertilizer. So this natural fertilizer will help the plant to grow better. Okay, so like this. So after turn to the ammonium form also can be absorbed back by some plants, right? But not every single plant can absorb ammonium direct to get the protein. Some that prefer to absorb is the NO3 negative, means the nitrate. Here also got nitrogen. This also ion. It's actually ion here. Soluble in water. They can be absorbed by majority land plant. Before come to the NH3, the intermediate is the NO3. Before come to the NO3, the intermediate is the NO2 negative. This is called the nitrate ion. I'll be like this. It's all the way underground. All this underground. Underground here. Okay, so all this underground, this one underground here. Okay, like this. So in the soil, we got the saprophytic bacteria and fungus, means this is a decomposer. Decompose the dead body into ammonium form first. Then in the soil, there's another bacteria here. It's called Nitrosomonas. Nitrosomonas species again. SP dot, you must underline Nitrosomonas. They can convert ammonium into nitrate. Then there's another bacteria called Nitrobacter in the soil. They can convert, okay, again a species, underline Nitrobacter. They can convert nitrate into nitrate. NO3 negative. Then from here can be absorbed by the normal land plant. This eaten by. Okay, so basically here already got 4 steps for you. Step 1 is the Rhizobium species. They can fix nitrogen for legume plant. Step 2, saprophytic bacteria. Number 2, this number 1, to decompose the dead body into ammonium compound. Step 3, Nitrosomonas convert ammonium into nitrate. Step 4, Nitrobacter convert nitrate into nitrate. And step 5, the last step here, okay, from nitrate also can be converted back to the nitrogen gas by this bacteria called denitrifying bacteria. It's at DE in front, denitrifying bacteria. So basically there are 5 steps all together. Every single step can give you 2 marks. Okay. For example, let's say mention, okay, the step from ammonium compound to the nitrate. You mentioned the bacteria is called Nitrosomonas species. Spelling correct. You underline, got the SP dot. You get 1 mark just for one name. And then you mention the function, just say convert from convert ammonium compound into the nitrate. Nitrate, NO2 negative. So like this, you get another 1 mark. So Nitrosomonas bacteria, you get 1 point. Function, you get another 1 point. So you get 2 points for every single step. And they will be all together, got 5 steps. So every single step got bacteria. You get 1, 1, 1, 1, 1, already 5. Everyone got their own function. So expand the function 1, 1, 1, 1, 1. So maximum will be 10. Okay, 10 marks altogether for the essay. In case they come out in the structure, they will give you maybe the whole diagram, but the bacteria not given. Maybe they point to a certain step, ask you name the bacteria. For example, maybe point to nitrogen to the legume. Is the Rhizobium species. Okay, depends on how they ask you during exam time. So basically this one is according to the overall. Please memorize every single step. Bacteria name, function. Okay. Okay, so we move on to the next one. Okay, for 11, can you explain about meiosis? Okay. So I give you a big intro one more time for meiosis. Okay, so basically meiosis is a type of cell division. It's a very special type of cell division in the body. Only our sex organ can perform meiosis, no other. Only our sex organ can perform meiosis. Your skin cell all cannot. All this only can perform mitosis. Only your sex organ can perform meiosis. And for boy, answer is your testis. For girl, is your ovary. So meiosis, anything what is so special about meiosis? Meiosis, the word meo means reduce. Because through meiosis, can reduce our number of chromosome into half. For example, we have 46 chromosome. Our body, every single cell, we have 46 chromosome. Then from here, through meiosis, come out the sperm is 23 chromosome. Yes, meiosis only got one purpose, is to form gamete. Gamete means sex cell. For boy, is sperm. For girl, is the ovum. So basically, meiosis only got one purpose, is to form sperm ovum. So only our sex organ can perform. Okay, so we start now the meiosis concept first. For meiosis, start from a diploid cell. Meiosis actually got 2 cell division. We name it meiosis 1 and meiosis 2. So for meiosis 1, means the thing divide into 2 cells, you will get both also haploid. And this is the main one for exam, is meiosis 1. Popular for exam about meiosis is also meiosis 1. After meiosis 1, the thing become haploid. Example, human, we got 46 chromosome. After meiosis 1, come out is 23 chromosome, already half. Okay, yeah. And then followed by meiosis 2, both also divide one more time, and you will get total 4 new cells, and all still haploid, still 23 chromosome. Let's say if the whole thing meiosis here, we talk about testis, result is sperm, sperm cell. 4 also sperm cell, and all 4 also haploid. Okay, so any difference between the diploid, haploid, haploid like this? Okay, example, we talk about starting this diploid cell got 2 chromosome. I assume got 2 chromosome. And these 2 chromosome, we name it as one pair of homologous chromosomes. Homologous chromosomes. Let's say it's one pair. Of course, for human, we should have 23 pairs of homologous chromosomes. But now, I only explain to you about the concept, so I only draw one pair. Okay, diploid. Let's say we assume that diploid equals to 2 chromosome. So when it comes to the cell division for this haploid cell, we get 1 chromosome is still do well. The X here also 1. So 2 chromosome is 2n. So 1 chromosome means half from 2. Answer is n. So n is 1 chromosome. Okay, yeah. And then for the meiosis 2, the X here is called sister chromatids. It will separate into, here we'll get half, here we'll get another half. So no matter is X form like this chromosome or the single form like this chromosome, this one and this one will count as one chromosome, one chromosome. So after meiosis 1, we get the answer haploid. After meiosis 2, answer is still haploid. So basically meiosis 2 is mitosis. Meiosis 2 is something like mitosis. Will be exactly the same like mitosis. Before, after, same number of chromosome. The very popular for exam is meiosis 1. Okay, let me explain to you meiosis 1 now. Meiosis 1 actually like mitosis. There are 4 phases. They got prophase, they got metaphase, they got anaphase, and also telophase, like pro, meta, ana, telo, PMAT. Just that for meiosis 1, when it comes to exam, okay, this topic, you must be very careful. If you talk about meiosis, you must say the prophase is 1 or 2. When you say prophase 1, you refer to meiosis 1. When you say prophase 2, you refer to meiosis 2. But if you just say prophase, behind no number, no 1 or 2, just say prophase, stop. This is for mitosis. So for meiosis, please write 1 or 2 to make it specific. No number, automatic, just prophase is mitosis. Okay, yeah. So I explain to you now is meiosis 1. So it's prophase 1, metaphase 1, anaphase 1, and the telophase 1. The most popular for exam is the prophase 1 diagram. Reason, crossing over. You can recall, what's your crossing over? Exchange of genetic material. So basically for crossing over here, okay, is the main point. So for exam, you have to focus more on the crossing over part. Okay, example. Okay, let's say for prophase 1 diagram, okay, you have to show the nuclear membrane disappear, use dotted line, standard. Centriole already moved to the opposite side of the cell, start to form spindle fiber. Like the normal prophase, nothing special here. Chromosomes also condense. Okay, let's say now I assume that only got 2 chromosome. Okay, only got 2 chromosome. I assume that diploid is 2 chromosome. 2n is 2 chromosome. But of course, exam normally should be 4. But I draw 2, just explain the concept. And I make it 1 black chromosome and 1 white chromosome. The black one represented say is the father, paternal chromosome. The white is the mother. So got 1 pair here. This is called homologous chromosomes. Homologous chromosomes. So basically it's 1 pair like this. So for prophase 1, the very special thing is, if you notice, the 2 chromosomes will come together, they will pair up. This pair up process is called synapsis. Imagine like two chromosomes. After they condense, they will come together to form one pair, one pair. This is called homologous chromosome. After form one pair, it's called homologous chromosome. And then the come together, the pairing process is called synapsis. So after synapsis, the two chromosomes will come together and they will touch each other. This touching point is called chiasma. So at the chiasma, there happened another very important event for SPM. The thing is called crossing over. The material, genetic material exchange, crossing over. Because crossing over is the main event that causes variation. Okay, what is the word variation? Means every human, we got different appearance, we got different genetic due to variation, due to crossing over. For example, let's say you and your brother or you and your sister from the same family, means same father, same mother. How come you and your brother or you and your sister got different genetic, different physical appearance? Answer is, for human, when we form sperm and ovum, every single sperm, every single ovum, they got different DNA. Why? Crossing over. So crossing over can cause variation, and this is very popular for our SPM exam. When they ask, okay, in prophase 1 or in meiosis 1, explain one important event, must be crossing over. How to explain crossing over? Exchange of genetic material between homologous chromosomes. Exchange genetic material between homologous chromosomes. Okay, and any result? Answer is, will form the new genetic combination. Any benefit? Variation. Means come out every single sperm, every single ovum got different DNA. Okay, so we continue first. After the prophase 1 diagram, it will be the next metaphase 1 diagram. For metaphase 1, you can show the chromosomes, the homologous chromosomes now line up, and with a big crossing over effect here. You can see the black and white mix already. That's the crossing over effect. Central, and spindle fiber are now attached. Actually, it's the spindle fiber that arrange the chromosome to line up at the middle here. This middle line of the cell is called cell equator. Of course, there's another name for the cell equator, is called metaphase plate. So cell equator can, metaphase plate also can. One more time. For the metaphase 1, how you explain the chromosomal behavior? Answer is homologous chromosomes, means one pair, line up at the cell equator. Okay, and then after metaphase 1, followed by the anaphase 1. So for anaphase 1 here, you can see the homologous chromosome are they separate and move to the opposite side. This one is called homologous chromosomes separate. And for the last one is the telophase 1. Just to complete meiosis 1 first here, telophase 1, you will see the two nuclear membrane reform here. Central remain, and for each nucleus we have only one chromosome. So ending is only one chromosome in one nucleus. Starting here got two chromosome, here got two chromosome in one nucleus, but here only got one chromosome in one nucleus, split into two new cell. Then here is actually called haploid cell. That's the reason why after meiosis 1, diploid become haploid. The number of chromosome divided into half. The main answer is the anaphase 1. This is the key explain why diploid after meiosis 1 will become the haploid. Main answer is anaphase 1, the homologous chromosome separate. That's why diploid become haploid. Okay, So I repeat here for meiosis, important point is only sex organ can perform. Number one, purpose is to form gamete. For boy is sperm, for girl is ovum. Okay, and then very important event in meiosis must be meiosis 1, must be the prophase 1 crossing over. If they ask how come the diploid starting after meiosis 1, we form the two haploid new cells. Diploid become haploid. Reason is anaphase 1 because of the homologous chromosomes separate. When you explain meiosis 1, the main keyword is homologous chromosomes. Of course, maybe from some reference books, you can see the word tetrads or bivalence. All this refer to the homologous chromosomes. They are the same. Okay, just the same thing. Okay. Okay, so this one is for meiosis. Okay, one more time for you. For meiosis, the thing to focus is about the prophase 1, drawing. Okay, sometimes in exam they will ask you, okay, let's say they give you maybe prophase 1 diagram, they give you the metaphase 1 diagram, but just that the anaphase 1 diagram is empty. When it's empty, you have to draw your own one. Means you have to know how to draw as well. For chapter mitosis, meiosis, you have to know how to draw for every single phase drawing. Number 2, explain the chromosome behavior. Number 3 is count number of chromosome. Let's say give you one diagram, they will ask, okay, for the diploid cell got how many chromosome? The haploid cell got how many chromosome? So basically, you have to base on the diagram to mention diploid got how many chromosome or haploid got how many chromosome based on the diagram given. Not based on human. Human must be 46. Sperm must be 3. But some say according to the diagram, so answer must according to the diagram given. You have to count from the diagram, not based on human, based on the diagram only. Depends on how they ask you. If they say human, 46 for diploid, 23 for haploid for sure. Okay. Okay. Next question. So it's based on the previous one. Motor neurons is efferent neuron. Answer yes. Okay, yes. Motor neuron, one more name is called efferent neuron. Okay, like just now, the input is the afferent, output is the efferent. So input is A, output is E. Input got one more name, it's called sensory neuron because the impulse afferent get from the sensory organ. So one more name for the afferent neuron is called sensory neuron because the impulse come from the sensory organ. But for the efferent, the output one, the efferent neuron, one more name is called motor neuron. Yes, because it send impulse to effector. Let's say to your body muscle for it to respond. You move like motor. You're moving like motor moving. So basically, the efferent send impulse to your muscle to respond to move, right? So like send to motor. So one more name for the efferent neuron is called the motor neuron. They are the same thing. And how to differentiate also afferent neurons? Okay, so it's based on this one. Okay, so afferent neuron, efferent neuron, interneuron. Okay, one more time here. Interneuron here with no myelin. Okay, they got no myelin, automatic is interneuron. If got myelin present on the diagram, you must look at the cell body, location, the position of the cell body. If the cell body is located at the end of the axon, is motor neuron, means efferent neuron. Motor neuron and efferent neuron are the same. If the cell body, the nucleus, located at the middle of the axon here, this is the sensory neuron or the afferent neuron. Okay, yeah. So I repeat here. Motor neuron and efferent, they are the same. Okay, and then the sensory and the afferent are the same. And for the word neuron, actually we got the E behind neuron. This is actually correct. This neuron is correct, but this is the US spelling. And this neuron is the UK spelling. So normally based on our textbook, if you refer to the BM version, This is the neuron, also for BM. But just that here, Malaysia, we follow UK English, so got the E behind here. Okay, for the neuron. So neuron, yeah, here got E for our syllabus. But for BM and US English, here got no E for the word neuron. Motor efferent, same. Okay, so we can revise the next one. Okay, can you explain about hormones in plants? This one is chapter 3, form 5 syllabus, the last part, plant hormones. So for plant hormones, actually, okay, our syllabus focus only on one. Is the hormone called auxin. Number 2, we focus less, is the one called ethylene gas hormone. So plant hormone, we focus more on the one called auxin. Because auxin can link to the phototropism. You can recall phototropism for the plant, then normally the shoot will grow towards the sunlight direction. This is called phototropism. Okay. Then for the root, we grow all the way ground. So we grow all the way underground. Okay, follow the gravity. Is called the geotropism. This whole thing is actually controlled by the plant hormone called auxin. Or make it simple for you, you can imagine the one called auxin, the plant hormone called auxin, is something like human growth hormone. We can grow taller and taller, we have the human growth hormone. Plant also got the plant growth hormone, but the name is called auxin. So basically auxin like the plant growth hormone. Plant can grow taller and taller, grow towards the light, grow towards gravity, all this controlled by auxin. Means auxin is something like the plant growth hormone. Okay, okay, let me explain to you about auxin. Okay, for auxin is the main plant hormone that we focus for the SPM syllabus. There's one short form is called IAA. IAA is actually also auxin. IAA is stand for Indo acidic acid. It's actually auxin also. So for this auxin hormone, let's say first we link to the phototropism first. Okay, for auxin basically is produced by meristem. Means at the tip of a shoot, let's say it's a shoot. At the tip of the shoot, here is the meristem, this region, meristem. Here produce the auxin hormone. Then the bottom here, this region is called zone of cell elongation. So the cell will elongate, will grow in size one. And then this is the auxin. I put IAA for auxin. This is auxin. Produced by here is the meristem. This is meristem. Or one more name for this zone here is called zone of cell division. So meristem here produce the hormone, and then this hormone can actually diffuse down. Diffuse all the way to the bottom to the zone of cell elongation. Okay, so auxin is produced by meristem. Here produce the auxin. But the target organ for auxin is bottom here, zone of cell elongation. Of course, the function is to elongate, promote cell elongation. So with more auxin come to the bottom, the whole thing will grow taller. Like this. Got auxin come to the bottom, then the thing here can perform cell division and also the elongation, so will grow taller. So like the growth hormone. Okay, special case. If this is the shoot and here got sunlight, when sunlight come from just one direction, one way, normally the shoot will grow and bend towards the light, and this is called positive phototropism. Positive phototropism. Okay, like this. Okay, it's about auxin actually. Let's say this is the auxin. Auxin here. when sunlight comes from just one direction, auxin is sensitive to light. Then the auxin, they will diffuse, they will move away from the light and then accumulate at this region that receive less sunlight, and then diffuse all the way to this side and then all the way down also. So when auxin accumulate more on this area, auxin is actually the growth hormone. So when this part of the cell got more auxin, the cell here can elongate faster. How about this region that facing the light, the brighter side here, that with lesser auxin, so will grow slower. When here grow faster, here grow slower, the shoot will eventually grow and like bend towards the sunlight direction and show you positive phototropism. So basically auxin is something like the growth hormone. The more auxin come to the cell, they will elongate faster. Lesser auxin, elongate slower. Okay, yeah. So just explain to you why the should normally grow towards the light because of the auxin. Okay, another one is the geotropism. Okay, let's say for the geotropism also can involve auxin. Let's say for a seed, you can see normally when you put the seed horizontal like this, horizontal, the tip here let's say is the future shoot. This is the future root. Okay, it's called plumo and the radical. So normally the plumo here can grow all the way up. Grow, put horizontal for seed, put horizontal, it will grow all the way up. Auxin control. Then the radical future root will grow all the way down, follow gravity, also auxin control. Okay, something like this. This is let's say for the seed, and then okay, here is the auxin produced. Due to the gravity, auxin will accumulate at the bottom. This side also the same. Seed. Okay, then with bottom here got more auxin, bottom here more auxin. Okay, with more auxin, let's say this is a future shoot. It will grow more. Here less auxin, grow less. So it will grow and bend up. Up against gravity, show you negative geotropism. Then here with more auxin, okay, more auxin bottom here, top less auxin. Okay, for the shoot, sorry, for the root is the opposite. For the root here is lesser auxin can grow faster. More auxin will inhibit, will stop the growth, inhibit the growth. Less auxin promote the growth. So it's the opposite. So when it comes to the opposite, one grow up, one grow down, also in opposite direction. The one that grow up against gravity is called negative geotropism, shown by the shoot. Okay, then the grow down one is the root, is called positive geotropism. Okay, so basically, okay, I hope all this can help you to handle your mid-year exam. Okay, and wish you all the best in your mid-year exam. So perform your best, do your best. Okay, and we will continue next Sunday, same time. Hope to see you again. Okay, so basically today we revised already got few topics: cell division, oogenesis, reflex, impulse, antagonistic muscle, and the auxin hormone here. Okay, if example about auxin hormone, normally is about this one. Okay, hope to see you next week. Thank you for watching. Do not forget to visit spmflix.com for more free online learning video. I hope you enjoyed this section. Tanya Cikgu will be live again next Friday, 3 p.m. See you again. Bye.